Am I turned on here? Yeah. All right, I feel like I trickle in for a while. I have sort of two parts of this talk, so hopefully part two will be more people here for part two. I'm Steve Andrews, and mostly thank you to Bill and Brian and Ilya, who's not here yet, and everybody else for sort of all the QBio organization, which has been really fantastic. It was a great conference, and glad to be here. I'm a staff scientist at the Frederick Research Center in Seattle, working with Roger Brent and his group, and looking at cell signaling in yeast primarily, but sort of more generally, mechanisms for high fidelity cell signaling. So, as you all know, I'm assuming, cell signaling, the kind of canonical picture is ligands bind receptor, receptor activated, receptor activates downstream proteins, turns on gene expression and responses. Responses are such things as cell differentiation, apoptosis, cell migration, immune responses, lots of kinds of responses. And if signaling goes wrong, you get wrong responses. You get cancer, you get diabetes, you get autoimmune disease. And so it's important for signaling to go correctly because you need to have the right parts in there. You need to have good quality signals to make sure you don't get diseases or problems. So with that sort of introduction, it's important for cells to signal accurately. Despite that, recent results suggest that cell signaling is noisy. I wish Ilya was here because he's a co-author on this paper. I kind of walked this paper by Chiang et al., Levchenko's group, published a couple years ago. They looked at signaling in the tumor necrosis factor system. So TNF, tumor necrosis factor, is the input signaling pathway. Output is NF-kappa B, which they quantified. So they experiment, mix together cells and signal. Signal means TNF. Image the responses, image the NF-kappa B output, quantify it, look at the distribution of responses. What they find is at low levels of input, of TNF, you get a distribution of output. More and more TNF, you still get distribution of outputs now for an overall higher value of output of NF-kappa B. But the whole way, there's a wide variation of outputs. So ask a question. Give an output value from a single cell. You have one image. How can you estimate input value? Can you, given you have, say, 10,000 units of NF-kappa B, how well can you estimate what input must have been? Well, it's one of these things up here probably. Probably was not down here. Or if it was 5,000, it's probably, well, one of these things here. But basically you can tell if it's high input or low input. You can't tell a whole lot more than that. So really you have kind of two states, high or low. That's all I can infer. So basically you have 1 bit or 0.96 bits of precision is really all I can infer from this experiment. They said from that, therefore, signaling is very noisy. Similar result from Uda et al. From last year. Different signaling system, now 3 different signaling systems, multiple outputs. They looked at multiple signaling pathways among these systems. And the key thing here really is the scale on these bar graphs. They still say 0 to 1 bits of information. That's all you get out, not a whole lot. So I did the same thing. I looked at yeast cell signaling. In this case, it's the pheromone response pathway. Input is yeast pheromone signaling pathway. Output is YFP fluorescence. Experiment looking at published data by Coleman Lerner et al. From 2005, kind of made sort of more available in some sense by Alan Bush et al. Last year. And so, same analysis, I call it population analysis. The coefficient of variation is 0.45. In other words, the standard deviation of brightness is here is about half of the mean brightness. That's our coefficient of variation. From that, I plotted a called dose response noise curve. X-axis is dose, y-axis is response. It's called dose response curve, shown in black. The noise kind of shows your envelope of responses. So input of 10, your mean response would be about 4, but your range would be from 2 to 6. And then from that, to math, out comes 1 bit of information. So yet again, 1 bit of information is in line with prior measurements. The question is really not how much I measured here, but how much does a cell actually know? So what we answered was, given output value from a single cell, how can the experimenter estimate input concentration of signal? We want to know, though, how much the cell knows. So 2 possibilities. One, signal is just plain noisy. Maybe Levchenko is right. Signaling is just plain noisy and it's imprecise. Or maybe measurements are actually good relative measurements. So a signal is quite repeatable, quite consistent from one measurement to the next, but cells are different. Some cells are high reporters, some are low reporters, but each cell individually is consistent one to the next. Perhaps also, if good absolute measurements, signal is consistent from one, was consistent, and also cells have internal calibrations. They say, I know I'm a strong reporter, and this is this high signal, therefore I have a calibration for the signal. So, first talk about relative measurements and argue basically, yes, these are actually good relative measurements. Then show actually absolute measurements are plausible. I don't have a good data there, but it shows plausible. So, first of all, yes, in fact, cells do cycle consistently over time. This is the same image I showed before from Coleman-Lerner on the right-hand side. Notice the bright cell here. It was also bright at 45 minutes, it was also bright at 15 minutes, it was also bright at 0 minutes. So it's consistently a bright cell. This cell is a dim cell. It's always been dim. That cell's bright. It's always been bright. So cells are consistent over time. But more quantitatively, this graph shows the fluorescence output as a function of time for many different individual cells. Each line is one cell, showing induction of expression at time 0. Basically add pheromone to the system, then watch the fluorescence increase over time. Again, you're seeing consistent trends. Some are high, some are low, but it's not random. To quantify this, I subtract off the mean for the whole population, divide by standard deviation, get nice sort of flat lines, well, wiggly lines. And then from that, find the standard deviation of each individual trace. And the average standard deviation was 0.32 on this rescaled coordinate system. So your single cell noise is 32% of the population standard deviation. From that, population C is 0.45 times 32% is 0.15 to a single cell coefficient of variation. So now here's my new dose response noise curve, now much narrower noise envelope, and I'm getting out 2.3 bits of information. So conclusion from here is no, cell signaling is not noisy. Actually, it's consistent from one to the next, at least 2.3 bits of precision in cell signaling. And that's the E system. And the same exact logic applies to other systems as well, so I'm sure they have also substantially higher information than they've been seen before. Next, going to absolute measurements. How do you quantify cell internal calibration? Simple answer is I don't really know, but I can tell you how I would do it if I were a cell. And the answer is gene expression is often correlated between different genes. So you might be familiar with Elvis and Swain paper on interdextrated noise. This is, once again, Coleman-Lerner et al. 2005, kind of similar to a project. They looked at fluorescence, CFP fluorescence expressed from a constitutive promoter. So you're always producing CFP. And then y-axis is YFP fluorescence, which you just look at from this whole signaling system. And each dot represents a single cell. In this case, a strong cell up here has high constitutive CFP expression and high YFP expression from the induction. This weak cell is weak in both ways. Immediate cells are immediate in both ways. So most cells tend to be sort of fairly closely aligned to this one kind of diagonal. So perhaps cells could use this as an internal kind of meter stick and say, I got this value of YFP that corresponds to this value of my internal known CFP. So this is not really true for the cell signaling, but conceptually for the analysis, I said, here's my input. It maps this range of outputs of YFP those range of outputs of YFP map to these range of inputs of YFP, map this range of outputs of CFP, and combine that now. Single cell noise is 0.15, that's up here. That's coefficient of variation. 0.38 is the coefficient of variation for the expression correlation. Combine those using the sum of the squares and square root then at the end. To 0.41 is our coefficient of variation for the kind of the calibrated signal. And from that, you get 1.1 bits of information out for this calibrated signal. So, conclusion of this little section of the talk, something like that is not noisy. You got 23 bits of relative precision, and you can at least get 1.1 bits of this absolute precision, of calibrated precision. I see at least here in sort of italics in this case, because that's just looking at a single kind of meter stick, a single constitutive rotor. If you had multiple ones, or cells with an average of all genes or all protein expression, you can maybe do much better than that. Obviously, you can't beat 23 bits, that's some upper bound, but you can do well more than 1 bit. Yes?
How long cells remember when you own the rest of the
Right. So are these independent measurements or not? Is what you're wondering, I'm thinking. You're saying, are these, so I'm arguing these are consistent measurements from one to the next. Are you asking sort of, are they independent measurements? Is that sort of the question?
Yeah. If you would be doing like, okay, you trace the cell, and you will be doing independent stimulations to the cell.
Yeah.
Okay. And let's say that the first stimulation, the cell was not sensitive.
Yeah.
So what is the chance that the cell will be not sensitive, let's say after 1 day or after 10 hours?
Right. That's a good question. I don't know the answer. But I'd like to do that experiment, do that over and over again, sort of give a cell some signaling, some input, look at the output, then turn it off again and try again. Turn it off again, try again. But it hasn't been done now, at least to my knowledge. So the best I have is this time series. I'm assuming each measurement is essentially independent of the prior one. But there obviously is a memory. I mean, for example, just the obvious one, YFP is being produced, not being degraded, and so you have a constant buildup of YFP from one time to the point to the next. So you have a memory in there. It's not really an analysis, but it's a good point. More questions? All right. Next topic. This is a kind of similar style of thing. It's still on cell signaling fidelity in yeast cells, but quite different sort of approach to it. Looking at BAR1 secretion by yeast cells. And I wasn't going to present at all, but because of yesterday's sort of discussion of M cell, I decided that I needed to promote my own work a bit more and let you guys know about this. But my software called Smalden, which I'll get to. So this is now, again, yeast cell signaling, the same basic system, but now looking outside of cells. Now we have 2 cells. Yeast cells have 2 sexes, an alpha cell and an A cell, mat alpha, mat A. Mat alpha releases a pheromone called alpha factor, and not shown here, but sort of good biological knowledge is mat A cells release pheromone called A factor. That's not really part of analysis, so it's not shown here, but good to know. Pheromone, or alpha factor, yeah, change pointers to when it actually works. So pheromone is released here. It binds to receptors on the mat A cell, and which binds it turns red in my picture. If the receiver cell, the mat A cell, takes enough pheromone, oh, that's fine too, thanks. Enough. If mat A cell takes enough pheromone, it will then grow towards the mat alpha cell, the emitter cell. and it might go wrong. If it doesn't sense it well enough, it might go in the wrong direction. It might think there's a mating partner, but there actually is not one. It might not mate when it could mate. So again, it sort of makes sense that cells would have good quality signaling here, because if they signal incorrectly, that will lose cell fitness, and that'd be selected against. So we're going to argue that there's a good reason to expect to be an accurate signaling system. Here's a paradox. It's a apparent paradox. Met A cells secrete a protease called BAR1. BAR1 degrades alpha factor, degrades the pheromone. You're thinking, well, why would the receiver cell secrete a protease that degrades a signal before it's even sensed? My little clip art thing says, so a monkey covering its ears to hear better. Why would you block out the signal to hear better? So, puzzled by that, and then simulation of that is my Smaldon software. So, well, software introduction first. Smaldon is a lot like MCell, which you've heard about in this class. It's a spatial Hutchinson simulation, does continuous 3D space, fixed time steps, diffusion reaction surfaces, and tomorrow I'll have a tutorial on that, so please come to that. Again, it's like MCell. Smaldon, by comparison, is more accurate. Smaldon's errors are at most 2.5%, whereas MCell is 200% error sometimes. Smaldon is about twice as fast, has a lot of other features, has, I think, easier user interface. It's an all text-based interface as opposed to a GUI. It's more cited than MCell. It's basically a little plug for Smaldon. I've written it myself, and it's a great software, I think. So I encourage you to use it. And most of all, don't write your own software, please. Don't write your own reaction diffusion software, because it's been done before, and this took me 10 years of work, so please use it. That's Smaldon. My model, I had one central receiver cell, a MAT A cell, 6 surrounding MAT alpha cells, which secrete alpha factor, the pheromone, of which one of these is a high emitter and 5 are low emitters. The question is, how well can the receiver cell find the direction of the high emitter alpha cell? And then they emit these little black dots are pheromone, green dots are BAR1, that's the protease, blue are unbound receptors, and red are bound receptors. There's about 10 to the 5 total molecules in the simulation. It ran overnight pretty easily. And a couple of questions are very simple here. Pheromone plus receptor forms a bound receptor. So things bind to pheromone, bind to receptors here. And green BAR1 plus pheromone is BAR1. That is simple degradation of pheromone. Procedure. I set molecular release rates. There was release rates of pheromone and BAR1, and one steady state. And then I look at the output. Output is this vector, which is called a binding gradient vector. Is what's the overall direction they use to tell sensing of the red dots versus blue dots, the bound or unbound receptors. A simulation, and what to do. So first, the black line is dose response curve with no BAR1. So with more and more pheromone release, you get more and more receptors bound. Makes complete sense. You add in BAR1 and you get a substantial decrease of response. So makes complete sense. You remove a signal, you get less binding, obviously. So little diagram, no BAR1, loss of binding, with BAR1, less binding. Here's the interesting one though. This is the binding gradient. The average gradient is the length of our binding gradient vector. No BAR1, it's a pretty low. With BAR1, you get much, much higher vector, this binding gradient. So why is that? So the logic, the thought process is, with no BAR1, you can easily diffuse from a starting point to the close-side cell, or it can only go to the far-side cell. Each one is just as likely, pretty much. So you get very little in the way of a preference to the left side. But with the BAR1 cloud protecting a cell, pheromone has low attenuation to the close side and has high attenuation as it goes through the whole cloud BAR1 to get to the far side. So you have a much larger vector for the binding gradient. So here's our direction sensing ability. Again, bottom is pheromone release rate. Here's average angle error. Black lines, no bar 1, is showing a fairly wide angle. It says we can't tell which direction the alpha factor is coming from. You add in bar 1, you get much lower angle error. So all of a sudden it sort of shrinks down this angle error, it says. It's there somewhere. Yeah. It degrades, it's just plain degrading, yeah. So reaction is bar 1 plus alpha factor forms of bar 1. Just plain degrades, alpha factor goes away in the model. No, it's unchanged. It's an enzyme. It enzymatically degrades pheromone. Yeah. Also, I skipped a lot of things in the model. So actually, bar 1 is also bound to the cell surface, which I did not include. That's more simplifications, but the basic story is like accurate and pretty simple. This kind of cloud of bar 1 degrades alpha factor. When I say that story. So, any questions there before I move on? Yes. How fast does it degrade alpha factor? Sorry. Yes. Is that steady state? Yes, these are all steady state models. And I believe so steady state, I'm guessing in reality too. So I started out with this model, and just ran to steady state. Ran until I got steady state concentration kind of pattern of pheromone, stage concentration of bar 1, and all these sort of leveled out in steady state. It went pretty fast. It was 10 minutes maybe of real time, maybe less than that, 5 minutes real time. I don't recall actual numbers. Yeah, well, it's sort of as a 1/r profile. And so it's high in the middle, then dies off as 1/r as you go away from the cell. So it goes out for a long, long way, but it flattens out. So the main challenge there is that it never dies out. It's always a gradient. There's always reactions that go farther and farther away. So the question is, how large summation volume do you include? And so I developed some new methods to deal with kind of how to trim things off. But also new method is hybrid simulation, but get to a different topic. Yes? If you increase the gradient of bar 1 you said? Okay, increase bar 1 secretion rate. Yes, just about much. Yes, certainly at low bar 1 secretion, you get low effect. If it's too much, you get all pheromones degraded. It's useless. And so there's a sort of sweet spot in the middle where the right amount of bar 1 gives the best angle error improvement. I didn't explore the bar 1 secretion rate value. I sort of chose one value in the middle and said, seems to work well. And basically, there's no data on actual bar 1 secretion rate, no data on what fraction of it is cell surface bound, what fraction of it is in the surrounding system. And also, this whole thing, I have to admit, is somewhat artificial system. It's quite artificial. Cells don't have 7 cells, nice little pattern like that. What you have is 4 cells in an ascus, it's called, and you have a membrane around that, and you have other cells farther away, or maybe cells kind of in other systems, but this whole thing is definitely an artificially simple system. That shows that to kind of show the idea of how bar 1 could improve signaling, and sure enough, it does well in this case. Yeah. Mm-hmm. That's a good point. Yeah. Yeah, that's a cool thought. Yeah. Yes. Um, right. This whole thing is non-equilibrium. There's constant carbon secretion. There's constant pheromone secretion. Binding is reversible to receptors. That's not an issue here. But I guess really it's the bar 1 degrading alpha factor. That's irreversible. Alpha factor is produced irreversibly. So that's probably your energy source for the whole system. All right, we have time. Let me just do a very, very fast mention the next topic and then conclude. So, yet another mechanism for improving signal transmission quality, dose response alignment. Again, yeast cell signaling system. And we observe at multiple stages in these pathway, at receptor binding, at G protein dissociation, at Fuz3 phosphorylation, at YFP expression, we see very similar dose response curves. Not perfectly, it's not exactly the same, but similar dose response curves. Just as I said, I wonder if those similar curves are leading to, or help improve information transmission. And the argument was yes, doing so avoids rescaling signals in the signaling pathway. Instead of saying, let's go from 50% down to 30%, up to 50%, up to 80%, up to 50%, it says, no, let's stay at the same ratio pretty much the whole way through. Obviously, it's not perfect. The screen one's a ways off, but it's pretty good alignment. So analogy there, say a photograph, if you rescale it, you lose information. I'll just stick over the analysis, which might say, yes, it works out more quantitatively as well. Here, place envelopes on these things. More careful picture with kind of inputs going to outputs. And then calculus of variation to actually prove and say, yes, in fact, dose response alignment does produce maximum signal transmission. So, conclusions. Cell signaling is not noisy. In fact, you get at least 3.3 bits of information of consistency. It can be calibrated to at least 1.1 bits of absolute cell signaling. By screening bar 1, yeast cells improve direction sensing. At least they could improve it sensing that way, they likely do. And small and useful utility research. And dose response alignment, information transfer by avoiding signal rescaling. And then thanks to coworkers, Roger Rent, my advisor, Electrical learner, a close collaborator, and other collaborators, and NIH and MITRE for funding. Thanks.